Hi everyone, my name is Chris and I'll be teaching you about protein synthesis, or at least the first two steps known as transcription and translation. Transcription is essentially the process of DNA being copied into RNA, ribonucleic acid. There are many varieties of RNA in which we will encounter, but specifically mRNA, or messenger RNA, is being made here. But why would we need to do that if DNA and RNA are so similar? Well, there are two key differences between them. RNA has a ribose sugar and uracil as one of its four bases, whereas DNA has a deoxyribose sugar and thymine as one of its four bases. Transcription contains three steps: initiation, elongation, and modification. First, initiation. Most of the process of transcription is done through an enzyme called RNA polymerase. This enzyme separates the bonds between the bases of DNA so that they can be copied into complementary RNA. In order to do that, it must first connect itself to a promoter where it attaches to DNA with a bit of help from a series of other proteins called transcription factors. A common one is the Tata box.
No, not that kind of box.
Anyways, the Tata box has these four nitrogen bases within DNA, signaling where the promoter is and where the RNA polymerase attaches. And this is why it's called Tata, because it's TATA. But anyways, it then moves on to elongation. This is done from a 5' end to a 3' end, which run antiparallel to each other within DNA. RNA polymerase moves along the strand, forming nucleotides that match up with their DNA halves. Cytosine matches up with guanine, and adenine matches up with Oh, wait. There is no thymine. Instead, uracil takes the two slots where thymine would usually go. And now you have RNA. Now to modification, and what I can basically equal to editing, like the 50 clips and takes it took for me to make this video. While DNA is copied from beginning to end, only certain segments are actually used later on. Those are called exons, which are expressed unlike their cousins, the introns, who probably just stay in their room and don't express themselves in a way that we can see them. They are cut out by something called a spliceosome. Think a glorified pair of scissors that removes the introns. See ya. This gives us a strand of mRNA. So, we have the pieces. Now, what in the world do we do with them? Well, we move to translation and make all of these bases into amino acids. That requires the help of another form of RNA called tRNA, or transfer RNA. tRNA has something called the anticodon, which matches up with the codons here from mRNA. These codons will be key to understanding what amino acids will be made from this entire process. There are also the two ribosomal subunits, the larger one and the smaller one, which help bind both the tRNA and the mRNA together. The first step of translation is initiation. Hey wait, wasn't that the name of the first step in transcription? Well, it is. Kind of. Also, the second step also shares the same name, but they have slight differences, so just hold it. Initiation in translation is done with the binding to a specific codon or trio of bases with the mRNA code of AUG, which is translated into MET. And no, not the museum. The amino acid known as methionine. This bonding is facilitated by rRNA, which is ribosomal RNA, and is the combination of the two subunits that I talked about earlier. The rRNA has three sections, the E, P, and A sections. The A sections hold the anticodon for the next set of codons that is passing through the subunit. The P section is home to what is currently being made, in our case methionine, and E is where tRNA goes once it's done. Next is elongation, which is the process of adding more amino acids to the chain of previously made amino acids. This chain is pretty short, so we only have methionine, but the way that methionine was translated, is the same process here, and in this case would make alanine. Eventually, each one is connected to each other by the process of hydrolysis and will make a very long chain. Eventually. So, how do we know which amino acid is made? Well, all we have to do is look at the chart. Besides a few exceptions, most of them will result in an amino acid. But for those exceptions, UAA, UAG, and UGA, they actually play a role in termination. Close enough. Termination is the step where a message is sent to make the protein chain stop adding to itself. Each one of these codons, instead of making an amino acid, it signals the process to stop by making a release factor instead, and the end of the amino acid chain is closed by a bond to water. And just like that, those are the first two steps of protein synthesis.